Hello, this is Mitchell Owens, and this is my extra credit video where I'm going to talk about the topic vaccination. And I chose this topic because I believed it was one of the more important ones and interesting ones to me that we went through in this course. So let's get started. To start off the discussion on vaccines, I would like to look at the different versions that we can see. The first one we're going to look at is the toxoid vaccine. And this type of vaccine is generated by curating a toxigenic strain of a virus. And then with that strain, we filtrate it many times and very efficiently. And this removes any residual proteins that it may have. With that, we're able to create a toxoid that's similar enough to the original that it can still create the antibody reaction that we're looking for in the body. This vaccine comes with many advantages for it. One of it is there's no reverse virulence. What this means is that there's no danger that we're going to get a reversion and that you could get sick from this type of vaccine. This vaccine also cannot spread to unimmunized individuals, so that adds some safety. And another positive is that it's stable in many conditions. And what this means is like how the vaccine can be in different temperatures, it can last a long time, and it can be stored in different ways. Some of the disadvantages that come along with this vaccine is that they can require an adjuvant and sometimes several doses as well to create a good immunity. They can also result in reactions in the local area that they were administered to. Some examples include diphtheria and tetanus. The next type of vaccine is the inactivated vaccine, or the killed vaccine. This type of vaccine results in a response from the body that's quite similar to the toxoid vaccine. Once the vaccine is administered, it can take around 14 days to actually build the response that would stop another infection. The response includes the correct Th2, which will activate the memory B cells and result in high levels of IgG in about 24 to 48 hours. Some of the advantages are the same as toxoid, which included no reverse virulence, cannot spread to other people, and stable in many conditions. But it can also include that the antigen includes all parts of the infection. The disadvantages of this vaccine are that it takes several doses, can result in some local reactions, inefficient, which kind of goes back to the several doses, and does not result in cytotoxic T cells. Some examples of these type of vaccines are the hepatitis A, flu, and rabies. The next type of vaccine I'd like to talk about is the subunit vaccine. And this one kind of contrasts with the inactivated vaccine because the subunit vaccine does not create antibodies for all antigens. Rather, these vaccines focus on antibody production for all specific antigens that are in the vaccine. We create these using proteins or polysaccharides. The proteins create a T-dependent response, like some previous vaccines that we've talked about. The polysaccharides create a T-independent response, where they migrate and encounter Th2. Th2 is not activated until the polysaccharide binds to a protein molecule. Some of the advantages of a subunit vaccine are the same as toxoid: the no reverse virulence, cannot spread, and also stable in many conditions. But another advantage is that we can also tell between vaccinated and infected individuals because they show different signs when you check them. Disadvantages include Again, some of the same with toxoid. Several doses can result in some reactions. Some examples of subunit vaccines include hepatitis B, HPV, and shingles. The last type of vaccine to be discussed is the live attenuated vaccine. This type of vaccine involves a weakened live version of the virus. We can grow this in foreign host as well as in artificial mediums. And because of how we grow it, the virus itself multiplies slow enough that it gives the body a good amount of time to develop a proper response. And once this response is created, it's able to be repeated when we actually get infected. Some of the advantages of this vaccine are that they require a small number of injections. Usually 1 or 2 can be good enough for lifelong immunity or immunity for a very long period of time. Some of the disadvantages is that it may cause an illness, and that's because the vaccine is actually alive, so there's always the chance that it could grow and multiply and actually cause the illness that it's supposed to prevent. Some of the examples of this type of vaccine include smallpox, the MMR vaccine, and chickenpox. Next, I would like to move on to one of the important elements that we can find within vaccines, which is adjuvants. Now, vaccines work by providing 2 types of signals for the immune system to operate with. The antigen is one, and this will guide the immune response to the correct pathogen. And the adjuvants activate the response through PRRs. The PRRs generate signals that are needed for the body to properly respond and recognize a pathogen. One of these such elements is the toll-like receptors that can work as sensors. In modern vaccines, now that we understand how adjuvants operate, we can make them specifically and manipulate them so we can get certain responses and results from the immune system. We can now make vaccines more efficient so they can result in longer duration, less doses, and get that specific reaction we want from the body. The future of vaccines is a very interesting and ever-evolving space. And one of the elements that is kind of pushing this change is our ability to genetically engineer aspects of antigens, and this allows us to make more specific, more effective vaccines and alter them in small ways to kind of tweak, test, and figure out what really works for us. Some of the new types of vaccines that we're heading towards include T-cell vaccines, which help generate some T-cell immunity, and mucosal vaccines, which help the mucous surfaces in developing immunity. And the last one is important because some of the vaccines that we've talked about are not typically administered into mucosal surfaces. So mucosal vaccines specifically can help areas that can also be affected by pathogens. Thanks for listening to my presentation on the vaccination topic. I hope you have a good day. This is the end of the lecture.